All right, good morning, everybody. We will get started with lecture 7 here. Um, and again, I apologize if there's been confusion about this lecture, when it was going to happen. Hopefully those of you who have missed it or got an incorrect Zoom link can see it on YouTube and all problems will be solved. Okay, so today we're going to talk much more about adaptive immunity, and then hopefully in the context of how it's relevant to the course. So we'll do that. My laser pointer. Okay, so we'll talk about the adaptive responses in the CNS. I think we'll focus a lot on this idea of antigen presentation and focus on this MHC class 1 presentation, MHC class 2, and how it sort of modulates Th1 and Th2 responses. So we'll talk a little bit more about T cell biology and B cell biology. And if you guys don't mind muting yourselves, I can hear some of you typing. I think I can mute all of you too. I'm not sure how to do that. Okay, so this is just a little overview. I think it's nice to look at in terms of the cross section between innate and adaptive. So let's just go through this a little bit. We've talked about the innate response. It's mostly going to be driven by myeloid cells, first lines of defense. Sorry, I'm hearing so much background. All right, well, I guess I have to deal with it. I can't figure out how to mute all of you guys. If you have it muted, please mute because I'm hearing you. It's bothering me. So let's go through this. So the innate is going to be a rather rapid response. It's going to depend on things like toll-like receptors, pathogen-associated molecular patterns. So these are going to be fixed responses. You add something that activates toll-4. There's a very sort of classic response that goes with it. So it's fixed. There's a limited number of specificities, and it's constant during the response. Now, as we learn about more adaptive immunity, it takes a longer time. So one idea, and the reason that we have an innate versus adaptive, is the innate gives you some time to build up that adaptive. So in response to making antibodies takes days to weeks, and the response is highly variable and has highly specific. And what happens is this very specific response improves during an immune response and helps you clear the infection. But remember, the innate and the adaptive, work in concert to clear infections, and I've shown you those graphs before. So we've talked about that this innate system complement toll scavengers, really kind of microglia and astrocytes. Now we have to talk about where these adaptive responses come from. So the antigen recognition molecules are something that we've talked about, these large glycoproteins called major histocompatibility complexes, MHCs, which are going to be on the antigen presenting cells, and they're going to bind to the T cell receptor, the TCR. All right. We'll also talk about antibodies, which look a lot like the T cell receptor, and that's going to be, from what's essentially the B cell receptor. Okay, so basic rules of antigen recognition are that antibodies recognize shape and therefore depend on preservation of the native structure of an antigen. The T cell receptor only recognizes peptide fragments of the antigen if bound to the self MHC. This is called MHC restriction. So you have to have the antigen inside the MHC for the T cell receptor to bind. And when you have this tight binding between the T cell receptor and the MHC, it really shows you that antigen processing has taken place. And we'll talk a little bit more about antigen processing. So these are the two MHC molecules, MHC1 and 2. And so I want you to notice a little bit differences about the structure. Okay, so MHC class 1 is made up of 3 alpha subunits, alpha 1 here, alpha 2, and alpha 3. There's only one transmembrane domain. So oftentimes when you look at immunology textbooks, you'll see it in this designation. There's just one transmembrane domain. Then there's something called beta 2 microglobulin, which basically stabilizes at the cell membrane. And so this represents the MHC class 1. And that you see the peptide here, the peptide here fits in this binding cleft. MHC class 2 is made up of 2 different subunits, essentially, or 4 different subunits, the alpha 1, alpha 2, and then beta 1, beta 2. And you can see there's 2 transmembrane domains. So oftentimes, like I said, in textbooks, you can tell them right away just from the illustration. It also has a binding cleft where you put the peptide. Now, what size of peptide that they present is different. So MHC class 1 is going to present things that come from the breakdown of proteins just in normal processing. So this tends to be about 8 to 9 amino acids in length. Okay, so that's for MHC class 1. For MHC class 2, it's what they call, it's like a sloppier presentation in that it doesn't matter as much. It can be about 13 to 25 amino acids long. So I mean, you're talking about a difference of plus or minus 12 amino acids inside that binding cleft. You can actually see that the peptide can kind of flop over the binding cleft. This binding is non-covalent, all right? And as we talked about, MHC is on all nucleated cells. So within the brain, we've talked about the plausibility of being on many different cells, including neurons. MHC class 2, remember, is only going to be on, MHC class 2 is only going to be on antigen-presenting cells. So there's really only 4 of them, B cells, dendritic cells, macrophages, and microglia. And we've talked a little bit about the difference between macrophages and microglia being more inducible antigen-presenting, not ones that do it quite a bit. Okay, so MHCs have what's called degenerate binding specificity, meaning they can put a lot of different things in the binding cleft as long as they're the right size. And then there's different MHC isotypes. Some are highly polymorphic. So we can see some different MHC isotypes that will lead to something called MHC restriction. So the variability here in the binding cleft is a little bit different between MHC class 1 and MHC class 2. So MHC class 1, again, has the alpha 1, alpha 2, alpha 3, and the binding cleft is here between alpha 1 and alpha 2. And this is where the kind of genetic variability is going to be, all right, in this cleft. And just to show you that MHC class 2, all the sort of genetic diversity here is in the beta 1. So this is where the different diversity comes in. Okay, so this is important, I think, to start to think about how you get this tight binding between 2 cells. So the antigen presenting cell is on top, all right? And what you see is that it has the MHC molecule. So on the left here, this is the MHC MHC class 1. You see the beta microtubulin. It has the antigen here in the binding cleft. And then you see that the T cell receptor sort of fits nice and snug against that. And then what holds it in place is, in this instance, is CD8. So you have that tight binding between the MHC and the T cell receptor. T cell receptor obviously on T cells. In this example, it's a cytotoxic T cell. So the major kind of locking protein is CD8. Over here on the right, you have an antigen presenting cell. This is MHC class 2. You can tell by the two transmembrane domains. The TCR is going to be on a CD4 cell. Again, it locks it nice and tight in place. And then it's anchored here by this CD4. So anytime you see a T cell or B cell or an antigen presenting cell interaction, you're going to see this type of arrangement. Without this, you wouldn't get that tight binding. So this is the T cell receptor. This is going to be the area that it binds the peptide, and this will be the area that it binds the MHC. The T cell receptor is, of course, located on T cells, and it's sort of in close proximity to these two other transmembrane proteins, CD3. So many of you probably know that you can look at pan T cell markers as CD3. And then when this interaction occurs, there's actually intercellular signaling that occurs as well. So there's a lot of diversity in the T cell receptor, and it involves recombination at the gene level. Talked about CD3. And it's also important if you think about antibodies, and those of you who've gotten the coronavirus vaccination, if you have antibodies against coronavirus, the antibodies actually look a lot like this T cell receptor, at least the top part. I'll show you that in a second. So this is, again, let's look down here. This is the T cell receptor that has the alpha and beta chains. And this is this red area is the region that's variable. This is where it's going to bind the antigen. And if you look, it looks a lot like this Y-shaped antibody. So the antibody here has two of these regions that look like the T cell receptor. Then it has a common chain. So this is called the
paracrine means it's signaling from one cell to the other. Autocrine means it's cell signaling.
Is that a question?
Acts on itself.
Did somebody ask a question or someone just mumbling out there? Okay. Anthony, I think your mic is on. Okay. So let's see. Okay, so the diversity of the T cell interactions is generated during development. And so let's take a close look at this picture, because I think the picture really shows you. So during development, progenitor cells give rise to a large number of lymphocytes, okay? And they do it just kind of within natural proliferation here. So if you look at these guys, they're each a different color, right? And you see that the antigen binding site is different, right? So this is all generated in development. So you already develop all these different potential clones. And this is so that if they ever come in response or ever come in contact with that specific antigen that they're built for, you see that you'll bind, and then you'll get proliferation expansion of that one clone, right? So diversity is generated during development. Then you have one specific lymphocyte that will respond, and this will proliferate. And you'll get a lot of clones against that same antigen. Does that make sense to everybody? So you've already developed all the T cells that you need. It's just whether or not they ever bind and find the antigen that they're looking for. Okay, so we've talked about this part already, this tight binding between the T cell receptor and the MHC, and then being anchored by the CD8 or CD4 proteins. We also require something else called the second signal. So this is called co-stimulation. Without co-stimulation, you get this what's called tolerance or anergy. So if you just have this interaction between MHC and T cells, you get no T cell response. So you need confirmatory factors that drive this response. So confirmatory factors, let's look at this side here first, the T cells, the Th1 and Th2 cells. So for Th1, which is the one that's going to drive a macrophage response, you need CD40 and CD40 ligand. So these are co-stimulatory molecules, okay? You also have co-stimulatory molecules for B cell interactions with the Th2, CD40, and CD40 ligand again. And then sort of the third signal are these cytokine signals that really either activate the B cell or activate the macrophage. And the cytokines here that are activating here are shown below. So to drive a B cell response, you're using things like IL-4, IL-5, IL-10, IL-13. And to drive a macrophage response, you're using interferon gamma, TNF alpha, IL-3. This is a little bit different. We'll talk about what this is, but this is actually an ARM T cell. And I'll show you what that means in a little bit. These ARM T cells will kill virally infected cells. Th1 cells will activate macrophages that express pathogen on MHC class 2. And Th2 cells, CD4 cells, will activate B cells that express on MHC class 2. So you want to know these different antigen presentation pathways, and I've tried to kind of put the yellow outline on them. So let's go through the steps that I really want you guys to know. So in order for to have antigen presentation, you need to have processed antigen. So this macrophage here has picked up something, it's processed it, it's put it on the surface of MHC class 2. Your T cell receptor is on a T cell. In this example is a Th1 cell, CD4 cell. The CD4 anchors it here. So you see that interaction that I've shown you a few times now. Now you have that important co-stimulatory signal. So that's number 3. This will be CD40, CD40 ligand. It's also called, or other ones are B7, CD28. You need this. If you don't have 1, 2, 3, you're not going to get a functional response. And the fourth thing you need is this cytokine signal. So that it's the fourth real thing that drives home this activation. In this case, to activate a macrophage, you're going to use interferon gamma. Does that make sense, everybody? It's sort of a big deal. Okay. So MHC class 1, let's go through the arming of a cytotoxic T cell. So this will be a viral infected antigen-presenting cell. This in my drawing is the shape of dendritic cell. It's going to present the antigen on MHC class 1. You're going to have your cytotoxic T cell with the T cell receptor and CD8 to form this tight binding. The co-stimulatory molecules in this instance are CD28 and B7. CD28 is on the CD8 cell, and B7 is on the antigen-presenting cell. So then the fourth signal here, the fourth signal here is the IL-2. And this is autocrine, which we've talked about, everybody knows, is that it's produced by the cell and the same cell responds to it. And you'll get the expansion of this CD8 cell. So this is the arming of MHC, or arming of cytotoxic T cells to a specific antigen. And why is arming important? So what we see here normally is our happy cell. The blue is self-antigen, and it would cause no immune response. There'd be no T cell recognition, no immune response. So this is normally what you have happen. Now, if this cell is infected by a virus, the virus takes over the cell machinery, all right? And so a virus needs to use host machinery to make protein. So as the virus hijacks these systems and makes protein, some of this protein will end up being degraded through the sort of natural cell processing systems. These degraded proteins end up in the ER, and then they get put on MHC class 1 just as a normal process. And now you present something that's non-self. So here's an example of that. You have your non-self viral protein here, peptide, which I talked about will be cut in about 8 to 10 amino acid fragments, peptides, sorry, and then put on the binding cleft. And now what you have is that ARM T cell. So in this instance, you notice that you don't need any other co-stimulatory molecules. This ARM T cell can just go ahead, and what it does is, from a few slides ago, and let me just go back so you can see it. I hate to do this, but The cytotoxic T cell releases FAS ligand, which binds the FAS receptor here, which all cells have, and it causes a very quick apoptotic response. So that's how that armed T cell will start killing cells. Okay, and we'll show this again in a minute, but this just shows you the processing. So for MHC class 1, like I said, the virus will take over host machinery and it wants to make proteins. And so it'll make proteins, proteins here that are degraded by the proteasome. All right. And then it chops it up. So anything that goes to proteasome is going to chop it up in the 8 to 9 amino acid peptide sequences. And then there's let me move my face out of the way here. Ugh. Okay, then it moves over here to the what's called a TAP protein, and the TAP protein is a transporter protein. It transports out of the cytosol into the ER. Okay, now move my face around again. Okay, so then here is the complex of MHC class 1. So it's inherently unstable. These MHCs are just naturally. So they need a number of support proteins. So in the case of MHC class 1, it's something called calnexin that kind of keeps it together before it has something bound in the cleft. And basically, if you don't have something bound in that cleft of either MHC 1 or 2, it's an unstable protein. So you have to keep it kind of stabilized before you get something inside that binding cleft. And so the MHC1 is pretty complex. It's almost like if you guys think of construction and you're taking a big dump truck and you're positioning it so that you can then take your backhoe and load a bunch of dirt in there. This unit's set up like that, and you have everything stabilizing the MHC class 1. And then here's our proteasome that's chopping up the proteins. In this case, it's that viral protein. We have TAP right here, that's transporter. It transports it out and it gets shuttled right into the cleft. Okay, and then as that stabilizes the MHC class 1, these other dummy proteins kind of fall away. And then you're left with what looks like a mature MHC class 1 with the peptide in the binding cleft, and then it's transported to the cell surface. So what cells in the CNS have MHC class 1? And pretty much all the cells that are nucleated have it. So all these cells really, really do. And the only reason that we kind of waver a bit is because in the medical school class, they teach you that oligodendrocytes and neurons don't have MHC class 1. Okay, so let's give an example where we know they definitely have MHC class 1, and this would be in endothelial cells. So let's go with a viral infection that gets into endothelial cells. So remember that to arm a cytotoxic T cell, it first has to be presented by an antigen-presenting cell. So this little star-shaped cell is a dendritic cell. It's going to express the viral protein on MHC class 1. Our cytotoxic T cell here is going to recognize that specific peptide through the TCR. And remember, this is where the variability is in the T cell receptor. Locked in place by CD8, you have the CD28B7 interaction, which are the costimulatory molecules, IL-2, which is going to cause proliferation expansion of that cell type. And now you have what's called an armed CD8 T cell. Now it's going to look for any more of that antigen bound to MHC class 1. And notice here that we have it on the endothelial cell. It doesn't need anything else. It's already armed. It just needs to bind to the T cell receptor. It's going to provide fast ligand, and it's going to kill that cell. And it's going to move down the line. So let's say the virus infected cell is right next to it. It kills this cell, comes down the next one, kills the next one. So you're basically destroying any of these virally infected cells in a chain. All right. And so there's examples of this in the CNS where you see arming of cytotoxic T cells against virally infected neurons. So this one example, this would be West Nile virus. You can see virally infected neurons that present antigen. to an armed T cell and get destroyed. Obviously, loss of neurons in West Nile virus is bad. You can also see astrocytes produce on MHC class 1 and be destroyed by armed T cells. Really, this table, I think, is trying to show you all the different ones that express MHC class 1. And again, it's on all nucleated cells, and I won't try to trick you with any questions about it, but for our class, let's say for sure that CNS macrophages have MHC1, microglia have MHC1, astrocytes have MHC1, endothelial cells have MHC1. And let's say that it's plausible that neurons and oligodendrocytes have it as well. Just know that there's some debate on this, but it's certainly, there are instances when you can see this happen in biology. It looks probably like these MHCs are a little bit more inducible on neurons than oligos and aren't normally expressed at high levels. Okay, does everybody understand that cytotoxic T cell? Great, because you're going to be testing on it. So the next one is MHC class 2, which again will be presented by a specific antigen presenting cell. And in this case, what we're going to see is that we have something presented by a macrophage. This is really going to be a Th1 response. So not only is it a helper T cell, but it's a Th1 T cell. You're going to see that binding that we've talked about. You're going to see the co-stimulatory molecules. You're going to see interferon gamma. All right, and this is going to activate the macrophage. So this is a Th1 response because it activates macrophages, and it's interferon gamma driven. If you have a B cell that picks up antigen, all right, and it's actually going to pick it up by binding to its B cell receptor and get internalized, processed, put on the surface. It's going to need co-stimulatory molecules. The key difference here is the cytokine profile that the T cell will use to activate it. And this will be from that intercellular binding domain, right? It's going to inform the T cell that it's binding to the MHC on a B cell. It's going to produce IL-4, IL-5, IL-10, and you're going to drive antibody production to that specific antigen. Okay, so this is processing from I hate Zoom. Okay, this is processing from something that's picked up by a macrophage. So let's say there's a bacterial pathogen, gets uptaken by the macrophage. It's going to be degraded in something called the phagolysosome, and this is going to be an enzymatic, acidic pH breakdown of particles. And when things get broken up in this way, this is where you get the kind of fragments that are larger, the peptide fragments that are like 13 to 25 amino acids. And it's going to be put on the surface of MHC class 2. And then it's going to be recognized by the specific clone of T cells that it was already generated to potentially find that antigen someday in life. Just like we saw for MHC class 1, that the processing is when what you do is here it's going to be different. So again, remember that any MHC is unstable. And so for MHC class 2, you have what's called the invariant chain, which keeps this guy stable. All right. And then it's pushed out from the ER into a vesicle. In the vesicle, it's like more of an acidic pH, and that invariant chain starts to break down. But it's important is that you have a little piece of protein called the clip protein that stays in that binding cleft. All right. You don't want anything getting in that binding cleft until it's sees a non-self antigen. So as that macrophage or B cell picks something up that starts getting degraded, it fuses just by chance with this vessel, and now you have the phagolysosome will change the pH again. This clip protein will actually fall out because the binding cleft opens up, and then during this opening, you have another sort of dummy protein that becomes associated with MHC so that it's stabilized. Again, anytime those binding clefts are open, these MHC molecules are unstable. And then as soon as that 13 to 25 amino acid binds in that cleft, then the dummy protein dissociates and then it's sent to the surface. And this is a comparison, so this is be a good slide to take a look at between the two different processing between MHC1 and 2. MHC1 is going to be just your normal degradation of proteins from the proteasome. That's why it's cut up in those base or the peptides of 8 to 10. And it goes the ER, binds MHC, needs stabilization, put out to the surface. MHC2, really the big difference is that you're taking things up in a phagolysosome, and then the vessels are emerging. You're getting that then stabilized and put on the surface. So this is a little bit about, why the peptides are different is because they're processed differently, right? So if you're processed by proteasome, it's put on MHC1. If it's processed by phagolysosome, it's going to go on MHC2. So what cells can present antigen on MHC2 in the CNS that we care about for neuroimmunology? There's another thing about antigen processing that's important is that if you need this MHC2 transactivator, what's called a master switch. So just because you might have the RNA for some of these genes, MHC, or maybe some of the cofactors, if you don't have this transactivator, this C2TA, you cannot be an antigen presenting cell. It's just not functionally possible. You can't do all the things that you need to do. So you can look for where this master switch is expressed in the brain, and it's only expressed on 3 cells. It's only on microglia, CNS macrophages, and astrocytes. So really, those would be the only 3 cell types that theoretically could present antigen. But that doesn't mean you can present antigen. So it just tells you that they can do the processing. And so let's look at this a little bit closer. So I think what's important here is to think about our CNS macrophages, which come from the bone marrow and really have a macrophage profile. And a macrophage is an antigen presenting cell. So does it have MHC class 2? Yes. Can it present self and non-self on MHC1? Yes. All right. So no surprise there. So let's focus on this right column. Does it have all the cofactor signals you need for antigen presentation? Yes. All right. So macrophage, for all intents and purposes, is a great antigen presenting cell. Now, if we look at microglia, you see it has a very similar profile with a lot of yeses, but you see this word inducible after it. So microglia have the capacity to express MHC and present antigen, but also self-antigen and viral antigen on MHC class 1. It also has MHC class 2 that's functional. It's much more inducible, meaning that you don't see it at high levels normally, but it can be induced in situations, and you'll see not only MHC2, but you'll see all the cofactors. When you start to look at the cofactor part for astrocytes, you find that there's very few of the important cofactors on astrocytes. And there's some evidence they're inducible. But remember that if you don't have all 4 of those signals, meaning you don't have the T cell receptor, you don't have MHC, you don't have the cofactors, you don't have the cytokine signal, you don't get functional antigen presentation. You get what's called anergy. So astrocytes really, there's no evidence that astrocytes can present antigen functionally. And this has been a big debate, over the years. I think the first classic paper that said astrocytes might be able to present antigen was in 1984. Showing again, for neuroimmunology, a lot of times people used the EAE or MS version of autoimmunity. They thought the astrocytes were presenting antigen, but this was in vitro, and there's just been, since this time, there's really no evidence that they present antigen, that they have the proper accessory or co-symmetry molecules necessary. So you're just really not going to get functional antigen presentation from astrocytes. So that really leaves us with just 2 cells that can express MHC and cause a functional response in the brain. And those are the CS macrophages and the microglia. So this was a science paper by a group at Dartmouth, Hickey, I think William Hickey and Kimura in 1988. And if you go look at this paper, I encourage you to do so because it's a science paper with, I think, 2 figures. And they were using bone marrow transplant studies in a model of EAE, which is the animal version of MS. And they're using the principle of MHC restriction and bone marrow chimeras, which I really don't want to take the time to explain the complex experiments that they use because they're pretty outdated and you wouldn't have to do that same experiment anymore. But let me just tell you briefly on what it means. It means that they were able to put in bone marrow derived monocytes that they knew would be self-reactive, meaning that they would cause an immune response against myelin basic protein. So in the animals that they did that for, they could see disease develop, okay? And they could track them essentially and know that those cells were here, okay? So they're first identify that these paravascular macrophages that we've talked a little bit about could present antigen, and they knew they were presenting self-antigen because it was causing this EAE disease, okay? They were using different labeling, and they could also see that there were microglia close by, but these microglia really weren't presenting antigen in the situation. So they're really the first to show that the paravascular macrophages had a full capacity to present antigen, which is a big deal in 1988 and why it was published in Science. Okay. So the fact that microglia have MHC class 2 expressed and they have that transactivator switch, does that really mean that they're competent antigen presenting cells? Because again, if you don't have all the proper co-stimulatory signals, you get what's called T cell anergy. So there are some situations that we've seen in vivo that microglia can present antigen and cause T cell proliferation. And this example is in Thayer's virus of MS. So Thayer's virus is something you can give to a rodent or to a mouse and is a naturally occurring autoimmune disease that develops. So viral induced mnemonic. And you'll see microglia present antigen in this way. The co-stimulatory molecule things looks like it's mostly inducible on microglia, meaning, they don't normally have it, but it can be induced. And then there's some weird stuff out there too, like you'll see MHC expressed in the white matter of brown Norway rats. No one really knows why, but you'll see it expressed. And you also see expressed under some inflammatory or injury conditions. Does that mean that antigen presentation is occurring? And that's sort of a question. Okay, so here's an example of when microglia and CNS macrophage likely present antigen, and this would be self-antigen. So this would be kind of building off what Hickey and Kimura showed, is that if you have self-antigen, this would be against myelin basic protein. So presumably at some point, the microglia and the CNS macrophage get access to this protein. It gets put on MHC2. And if they ever come in contact with a T cell, you could have this autoimmune response against self-peptide. And so in both cases, this would be a Th1 response. You'd be activating the microglia or the CNS macrophages using interferon gamma. Again, you have all the cofactors, you have everything you need to cause functional response. And this is just showing you, this is that paper from Thayer's virus in 2003, showing that what they're looking for and what they're showing you in the slides is that to cause functional antigen presentation, you can look at responses. So you should see two things. You should see those T cells proliferate, and you should see interferon gamma production. And what they did here is they compared splenocytes, which have loads of antigen presenting cells, to the microglia, which are the glia in the middle, to the macrophages, which are the CNS macrophages, or I think these actually might be bone-derived macrophages. But nonetheless, they found that of the three, the splenocytes, where they have really large number of antigen-presenting cells, could do functionally what they're supposed to do, cause T cell proliferation and cause interferon gamma production. And then you also see that the macrophages do the same thing, and that the microglia could do it, maybe not at the same level, but functionally could present antigen to cause T cell proliferation and interferon gamma production. So some of the first evidence of that. And so this is looking again at a chart of macrophages versus astrocytes versus microglia in the ability to present functional antigen on MHC class 2. Okay, so first of all, you have to be able to process the antigen. All right, you have to be able to get it into your cell. And again, let's look at the macrophages here. Hopefully isn't totally obstructed by our faces here on the Zoom call. But the macrophages, if you look at all the important things associated with picking up antigen, phagocytosis, having FC receptors, which will bind complement bound things. Can they process the antigen? Yes to all those, right? Makes perfect sense. Astrocytes really have very little ability to pick up antigen. Some evidence of phagocytosis, but no FC receptors, no ability to really process antigen in the same way, meaning that they're probably never going to be functional antigen presenting cells. Microglia, again, it's a lot of yeses, but it's more inducible. So you don't normally see all these things, but you definitely, I know all of you probably have seen examples of microglia using phagocytosis. We know they have FC receptors, and we know that they can process antigen. So the next sort of question is, can you kind of cause the proliferation of what are called naive T cells? So T cells that haven't seen antigen before. And again, start with the macrophages. The answer is yes. Does it cause IL-2 production? Yes. Does it cause Th1 differentiation with interferon gamma? The answer is yes. So turns out the microglia, a little more controversy here, but it's been shown to do all these same things. All right, so what I'm doing with this chart is summarizing loads of data for you, rather than we don't have going through individual papers, but just trying to summarize what the overall effects are. Again, this macrophages are baseline, so it does all the things it's supposed to do associated with an antigen presenting cell. Then this is something called restimulation of T cells. This is when a T cell has already seen antigen, but needs that extra kind of boost. Like let's say you see a T cell gets exposed to antigen in the lymph nodes, right? And then it goes out in circulation looking for that antigen. It really needs to see it a second time to kind of drive that signal home. So this is what it's looking at, this restimulation. And microglia can do all these things as well, just like the macrophage, and astrocytes cannot. So if I ask you any questions about astrocytes being a functional antigen presenting cell, your answer should be false. Okay, so write that down. They're not functional antigen presenting cells for MHC class 2. But what if I said MHC class 1? If you don't know the answer, make sure you do. Okay, so this is just going through the different cell types and sort of the functional response. And again, my face is in the way here, so let's get rid of that. So in the normal CNS, our antigen presenting cell, like our microglia, really don't express anything. If they come in contact with a T cell, there's no response, right? If some situation happens, let's say we have a viral infection and we start to see cells increase MHC class 2 expression. If the only thing they express is MHC2 and you see a T cell, what's going to happen is energy. You don't have the co-stimulatory molecules. You don't have that cytokine signal. You're going to get energy, meaning a non-functional T cell response. All right. And so if we ever see astrocytes in this way, this is what's going to happen. Just because they have MHC doesn't mean that they actually can do anything. Now, what cells can cause this functional activation? Meaning they have MHC2, they have accessory proteins, they have cytokines, and causes full functional response. Those would be the microglia, CNS macrophages, and dendritic cells. So relevant to neuroimmunology, these are the only antigen presenting cells that can present to a T cell on MHC class 2.
I have a quick question.
Sure.
Is there any like subsequent cell signaling that occurs in like a context of anergy where you would have like MHC2 presentation, but not, all of the other co-stimulatory factors and stuff? Like, is that involved in any other cell signaling events?
Yeah, I mean, certainly. So that would be important for if you, like say MHC class 1, if you're just presenting self antigen and you happen to have a T cell bind to you, it should cause anergy because you don't have all the rest of the costimulatory molecules, even if you did recognize self, right? So there are those checks and balances, and that's why it's there. So you're not having, just arbitrary immune responses all over the place. That's why you have all these checks and balances. It takes 4 precise signals to drive that response.
Gotcha. Thanks.
Okay, so here's a little comparison again that I think is an easy for review. What cells have MHC? What cells have MHC, 1 and 2? Basically, all of our nucleated cells in the CNS, we say have MHC1, and we'll give the med center its due and say it's plausible for neurons and oligos. And we'll say that only CNS macrophages and microglia have MHC class 2. Okay, everyone else will be no. And the reason these plus marks are here is because if you were to look at them side by side, like at a per cell basis on their ability to cause functional responses in T cells, which one's better, do you think?
Macrophages.
Exactly, right? So anytime you compare a CNS macrophage to a microglia, it's much better at those adaptive immune functions, highly more efficient. And that's what they're built for, all right? So microglia have the capacity to do it, but it seems to be very context dependent and way more limited. So we see a lot of these immune adaptive responses in autoimmune disease, right? So there's been a lot of work in neuroimmunology for decades now on using this model of EAE, where you get, MS-like pathology, multiple sclerosis-like pathology in the brain and spinal cord. And so there's been a lot of neuroimmunology work done here. And so what this paper was, this is a famous paper by Frank Hepner, published in Nature Medicine, and they use a model of MS using MOG. It's another myelin protein. So they immunize against MOG, you get self-reactive T cells that are going to cause pathology against myelin. And what they did here is, this is pre-Plexcon days where you can eliminate microglia a little bit easier. They had this construct, which is a CD11b herpes simplex virus thymidine kinase construct. And what you can do here, and you can still use these today. So you would add ganciclovir, and I think they did this systemically, and any cell that has a CD11b promoter that binds to the ganciclovir is going to cause a thymidine kinase induced cell death response. So in this model, you can functionally eliminate microglia by giving giving ganciclovir to the mice. All right, so think about this experiment. If you did it just with Plexcon, you could eliminate all the microglia, right? So this is predated that, but I think the effects are pretty similar to what they found, is that when they eliminated microglia, they saw less CNS pathology. So I'm just showing you some of the data slides. This is showing you the brown staining here is supposed to be sort of microglia reactivity here in the disease. You see it, and then in the microglia knockout, you really shouldn't see any microglia. And then they look for another number of flow markers and the infiltration of T cells and cytotoxic T cells, and see less if microglia had been eliminated. And then you see less pathology. So you see less IBA1 reactivity, less myelin damage, and more exon integrity. So they're saying that microglia have a role in this pathology, which has been followed up, many different times, that microglia are part of the pathology associated with these autoimmune disease. So let me show you a little bit about how that might be, and putting together the data that others have done in these MS models. And so there's a little bit of immunology here for you to digest. But let's say you do have come in contact with self-antigen. And sometimes it could be from a viral infection like Thayer's virus, where it sort of mimics it, or it could be from damage. During head injury or spinal cord injury, you often see the development of sort of self-antigens. So what would have to happen is that you would have likely these CNS macrophages pick up self-antigen like myelin basic protein. All right, and then as these surveying T cells come through, they're going to come in contact. And in this case, this T cells wasn't eliminated in terms of being able to self-recognize self-antigen, or it could have been put there on purpose. So you can do that in these models as well, put self-reactive T cells in there. But the binding is going to happen within this perivascular space. So this is going to be activation of a naive T cell. It's going to get activated. You're going to see some T cell proliferation. You're going to see chemokines produced. It's going to bring more T cells. Then we need to get past this perivascular space, which is the glute-limitin, right? So you're going to see these macrophages also digest that space using metabolic proteases, MPPs. And you're going to see then that the T cell comes in. And so let me get rid of my face again. And here, this is where the microglia likely come into play, is that once that T cell gets through that barrier, it's going to be looking for this MHC again for co-stimulate or re-stimulation. This is likely where those self-reactive microglia come into play, would also have that same antigen. Now you have a highly activated T cell producing cytokines and a highly active microglia producing cytokines. And in this example too, you can get these highly active microglia macrophages that can actually physically strip off the myelin. All right, and there's been some high impact papers showing that as well. So this is kind of like putting them in context and showing you a little bit of the space and variation between the CNS macrophages and the microglia. So if we take a look here, this is the idea of indirect demyelination. So you have the activation of microglia, macrophages, and T cells. The cytokine profile here is going to be high in TNF alpha and nitric oxide, as well as interferon gamma and IL-1. So this is going to be a very strong, what's called Th1 response, right? So these cytokines can damage the oligoprogenitor cells. So I think all of you, probably from Dana McTighe's lab and others, know that TNF alpha and nitric oxide can kill these progenitors. So you're not getting that remyelination. They can also damage the oligos that are wrapping the axons, where you lead this demyelination. So this would be kind of indirect demyelination caused by these inflammatory cytokines and reactive mediators. Okay, so just a little side note here. Does MHC represent pathology? We'll talk about MHC being increased on microglia with age, and we'll talk about a little bit what that means or what that represents. We know that these MHC2 positive microglia also are the ones that surround the beta amyloid plaques in humans and in rodent models. We also see MHC around CNS pathology, around things what's called Bulgarian degeneration and trauma. And we can see it sometimes in the absence of infection during repair processes. So I think it's important if you do come across MHC in the literature being increased in certain situations, you have to put it into the context. Okay, so for the last part, and I don't really know how long I've been lecturing for. It looks like I've been lecturing for about an hour. So just have a few more slides, so we'll just get it done. So we want to talk a little bit about B cells in the remaining time, and so let's do that. Okay, so CNS infection, you have antibodies against an extracellular pathogen, and then we also see antibodies in autoimmunity. So this could be MS again, or this could be lupus, where you have antibodies against self-antigen. In lupus, it's DNA, and then in MS, EAE, it's in some of the myelinating proteins, myelin-basic protein on oligos. We also see that you get antibodies produced in traumatic brain injury and spinal cord injury. And I know Phil Popovich's lab has shown a lot of work with B cells being found at the site of the injury of spinal cord. And B cell knockout mice have smaller lesions. So we think that the B cells and the antibody responses have some role in pathology. There's a disclaimer here at the bottom, MS and EAE, which we'll learn about from Amy Levitt-Racki when she comes to talk, is really a T cell mediated disease, meaning that I could take reactive T cells from one animal, inject them into another animal and get disease. You can't do that with B cells. But nonetheless, B cells seem to contribute to pathology because you have these autoantibodies. And if you take serum from EAE mice, and put it into the spinal cord, you see demyelination. So certainly some evidence of the B cell here being involved. I'll keep moving my face around here. This is the B cell receptor, which I told you before looks a lot like the T cell receptor and antibodies. So if you think about it, what you see is this is the pre-B cell receptor, and it's held together by these surrogate light chains. So these are huge globular proteins. There's two identical light chains and two identical heavy chains. So the heavy chains here are these. These guys here are the heavy chains. Here, the ones that have the transmembrane domain, and then the light chains are covalently attached to them. And the diversity here is going to be in the B cell receptor. These portions here, up here, the binding sites are going to be where the diversity is. This is what's called, the common part of the receptor. Okay, so just like the T cell that I showed you, that slide, that you get these different pools of T and B cells in development. So you get all these clones developed, and then hopefully you eliminate any of the self-reactive clones, but you're left with a huge diversity, okay? And just like the T cell, the B cell receptor recognizes specific antigens on MHC class 2, and you have tremendous diversity in the B cell receptor, and it involves recombination. So this is just giving an example that if you have pathogen, you're going to have recognition of both B and T cells. This T cell clone recognizes this part of the pathogen, this B cell recognizes this part, and you'll see expansion of both those B and T cells. So the big thing about B cells is that they get converted. Okay, so if the B cell is the antigen presenting cell, it has all the same things that we've talked about before. It has the MHC on the B cell. This is MHC class 2. It has the CD4 from a Th2 T cell receptor, the cofactors. This is going to be showing you two different versions of cofactors, CD40 ligand and CD40, as well as CD28 and B7. The cytokine signal here is going to be in the IL-4, IL-5, IL-6 family of cytokines, and you're going to cause proliferation differentiation. So what you're going to do is you're going to convert this B cell to what's called a plasma cell. And the plasma cells really only has one goal, and that is to massively produce antibodies. And it'll be antibodies that are specific to whatever is presented here, this specific antigen. So they look different and they have a different profile. So now if you see a plasma cell, there's no MHC expression anymore. It's just producing high rates of antibodies. There are different types of antibodies. Probably IgG is the one you hear about most. And IgG antibodies are involved in neutralizing toxins, okay? They're involved in opsonization, which we've talked about before, is coating a protein, mostly that's been, probably has complement on it. It can, let's see. What else does IgG do? So IgG gives you a lot of sort of the immune protection. If you want to take out you want to look at some other ones, IgE is the one that's involved in sensitization of mast cells. So these will be the ones that sort of cause that allergy type response. They have different properties as well. I think when you're first born and you get the mother's milk, right? You get a lot of IgA antibodies because it can cross and then transport across the placenta. Okay. It's mostly the IgG. Sorry, my phone is ringing. It's distracting me. Let's see. So this is the antibody again. This is the structure here. Is that Y structure? This is the heavy chain, two heavy chains and two light chains. Where the antibody binds is here. Or sorry, where the antigen binds is here, and this is highly variable, and these are the constant regions. The variable here is in red, and the constant is in blue. Okay, so this shows you a little bit more about the plasma cell. So your resting B cell has surface IgG or Igs. It has surface MHC. It does not produce antibodies. Um, it can switch isotypes. It's inducible growth, proliferation. And then a plasma cell has nothing except be able to produce antibody. And you can't really isotype switch. Isotype switch is when it switches the type of antibody it makes. And once a plasma cell is directed to produce antibody against a specific antigen, it just pumps them out. So you can see how it becomes very, very specialized very quickly. So from B cell to plasma cell. What do these antibodies do? Okay, so this again is going to be a Th2 response. It means that when you have activation of B cells, you're going to produce antibodies that neutralize bacterial toxins. So this would be mostly those IgGs. And you'll see it here. This is an example of neutralization. So you have this bacterial toxin, you have an antibody against it, and it form these kind of complexes that protect the cell from responding to the toxins. Also, we'll see this idea of opsonization. This is going to involve the complement system, where you're going to have binding to complement for degradation. You can also just opsonize a bacterial particle with antibodies. Either way, complement, non-complement, it targets it for ingestion and destruction. So you're going to have coding, activation, complement systems. The other thing you can have is activation of natural killer cells and activation of mast cells. So basically for antibodies, it's to neutralize and opsonize. This is getting back to something that came up a few minutes ago. I showed you this idea of autoimmunity with MS and demyelination caused by cytokines. So you can also imagine what the role of B cells could be by direct demyelination. And this would be plasma cells making antibodies against myelin-basic protein, or MOG, to oligoproteins. And then you have them opsonizing oligodendrocytes. So now you're coating an oligodendrocyte with antibody, which then the FC region, so this is the common region. Does anyone know what an FC receptor binds? The common area of the antibody. And this allows for macrophages or microglia to pick things up that are coated. So now you can have actual directed killing of oligos by immune cells. So on the left here, examples of indirect demyelination caused by cytokine responses mediated by T cells, microglia, macrophages. And here would be an actual direct demyelination caused by antibodies. And there's evidence that both of these parts of pathogenesis are happening in demyelinating disease. So that is the end of today's lecture. So stop sharing. And in this extra time here, if we have any, I know this is probably the one lecture that people may have the most trouble on, because it's much more immune based than we've done before. So if you have questions, now's probably a good time to ask them.
Yeah, I have a question going back to the Hickey paper, I think it was, with the EAE. Where would the macrophages pick up MBP if they're out in the perivascular space and astrocytes can't present antigen in that way?
Well, they made bone marrow chimeras with what are called self-reactive T cells. So they made chimeras with the T cells already kind of pre-selected that would activate a self-presenting self-antigen. So what really comes down to the T cell, as long as the T cell is surveying, meaning it goes into some of these locations, it's going to come in contact with either the meningeal max, the perivascular max. So that's probably the key, is that you have self-reactive T cells interacting with the with the bone-derived macrophages. So the experiment, I think, was that they could differentiate the bone marrow-derived monocytes because of how they put them in there, made chimeras, so they could basically know of that interaction occurring and where it was occurring. Nowadays, we'd use like fluorescent, bone marrow chimeras, so you could see the monocyte in the perivascular space. So it was a little bit harder for them to do it, but the results are the same. But yet, astrocytes for sure do not present antigen. It's going to be that interaction between the T cells that get into those spaces by surveying and the macrophages are already there. Remember that the macrophage or monocyte populations in there get turned over, so that they only last there a few weeks before they're turned over. So if you even, let's say you had normal, healthy, non-reactive and presenting myeloid cells starting there, and then if you did bone marrow chimeras and put in a bunch of reactive cells, over time you're going to see those monocytes within the perivascular space, meninges, other places. And then once they have contact with those T cells, that's when you can get that initial response. I'm not going to ask any questions on that paper. I was just showing you that because the paper is pretty complex if you look at it. Because we don't think in that way anymore. We would do it totally different and easier.